The title of my talk today is Resident Memory T-Cell Immunosurveillance of the Oral Mucosa and Periodontium. And this image will come up again a little later in the talk, so I'll explain it at that point. Looks like I've got to do this to now get this going. Okay. So before I get started, I just want to acknowledge some of the folks that have helped with the work. Of course, the Mass Disease Lab for their support. And specifically, I'd like to acknowledge Vineet Jog and Mocha Scott, who have been instrumental in some microscopy and RNA sequencing analysis, and also a very talented DDS PhD student at the University of Minnesota named Flavio Saavedra, who has helped with some of the ligature-induced periodontitis experiments I'll show at the end, and then, of course, acknowledge my support. And as was alluded to by Yina, this is a bit of a homecoming for me. So I grew up in Northampton, New Hampshire, and attended the University of New Hampshire for undergrad, and of course here we are today. And so a lot of the work that I'll be showing today is from a paper that's currently in revisions of the Journal of Experimental Medicine called Depleting 103 Positive Resident Memory T Cells in Vivo, Reveals Immunostimulatory Functions in the Oral Mucosa. So just to give a quick introduction to the talk, I'm going to try to cover a lot of ground here, but all thematically related to resident memory T cells, or TRM. So I'll first give an introduction to TRM to get everyone up sort of on the same page, and then I'll provide some rationale for why I became interested in studying TRM in the oral mucosa. I'll talk about how we generate tractable populations of TRM in the mouth to manipulate and study, and where in the mouth they're localized. I'll focus on the sort of middle and latter parts of the talk on the buccal mucosa or cheek, specifically what are the consequences of TRM reactivation in this location. I'll introduce a therapeutic strategy that we've developed for eliminating 103 positive TRM in the mouth, and then close with some tools and the initial independent laboratory focus. So what are TRM? So I find that immunologists love to use militarized references, or analogies, so I'll sort of start with one here. So this is, of course, a map of the United States, and I've overlaid a map of the U.S. interstate system here. So imagine that you're a general of an army tasked with sort of defending this vast space. One reasonable strategy would be to position, troops at sort of the confluence of several of these interstates, so that if you were positioned in Denver, Colorado, for example, you'd be able to sort of easily determine what was going on in the neighboring states of Wyoming, Nebraska, Kansas, New Mexico, and Utah. And when we consider the immune system and the circulatory system specifically, the strategy is what really has sort of evolved. So instead of surveilling all 40 trillion cells of the human organism, naive and circulating memory T cells really limit their immunosurveillance range to secondary lymphoid organs, that is lymph nodes and spleen. And in doing so, they're able to rapidly detect rare antigens. But there's sort of a problem in this system, inherent in the system, if it were the only game in town, and that is that infections do not spontaneously appear in lymph nodes and spleen. They occur on our skin and on our mucosal surfaces, and only later get into the draining lymph nodes. And so, sort of to return to this analogy, if you knew that your enemy was arriving by boat, you would want to redeploy your troops to the coast. And this is essentially what resident memory T cells, or TRM, provide: a mechanism for stockpiling specific immune subsets in barrier tissues that are commonly exploited by pathogens as portals of entry into the body. And so in these locations, TRM perform what we refer to as sensing alarm functions. So I'm depicting them here in the skin. So these are the TRM in purple here. And the important point to note is that these are really sentinel cells that are juxtaposed to these barrier surfaces that, again, pathogens sort of exploit as portals of entry. And upon secondary infection or the sort of stimulation through antigenic peptides, these cells are really the first responders, and they're able to rapidly intercept an infection or detect antigenic peptides and release a series of pro-inflammatory molecules, which collectively operates to orchestrate this local collaborative immune response. And this sensing and alarm program accelerates pathogen detection and clearance, while blocking subsequent viral spread. So residency is by definition a migration property, and so the gold standard approach for defining tissue residency is this approach called parabiosis surgery. So just to highlight how this works, this is sort of a hypothetical example here, but imagine you had two mice that were infected with the same pathogen. One mouse had cells that were red in this example, and one cell had immune cells that were blue. Of course, in practice, we use mice that bear unique and genic marks. But after the immune response evolves, if you were to surgically join these mice via parabiosis surgery and wait an additional 2 to 3 weeks, you'll get chimerism of the circulating immune system, such that if you were to look, for example, in the spleen of this original red host, it would be a 50-50 mixture of cells derived from the red and the blue host. But if you looked in a non-lipoid tissue, say the oral mucosa, for example, a little bit of foreshadowing there, the immune cells there would be derived entirely from the original red host. And so we've used this this procedure of parabiosis surgery to really demonstrate the predominance of residency across almost all of these non-lipoid tissues that we've analyzed. This is after systemic LC&B infection. However, these initial studies that were performed back in 2015 did not extend into the oral mucosa. So parabiosis surgery is kind of a cumbersome experiment. It's not always technically possible, and this has sort of obviated the use of cell surface markers as diagnostic of tissue residency. And so here we're looking at an antigen-specific CD8 T cell population that's been isolated from 2 different locations. In the blue traces the cells that have been isolated from the intestinal epithelial lymphocytes of the gut, and in red are cells that have been isolated from the spleen. And we now know that these 2 different populations represent resident memory T cells in blue and circulating memory T cells in red. So you can see that they're very phenotypically distinct, and these are some of the canonical markers that are used to identify tissue residency. So TRM are CD69 positive. They tend to be low for Ly6C. They express CD103, integrin beta 7, and they tend to be low for CD62L. And personally, I prefer the combination of markers, expression of CD103 and lack of expression of Ly6C, because one marker is upregulated and the other marker is downregulated, and I think there's sort of some additional power in that. So before I jump into the main sort of body of the talk, I alluded to some of the work that I did early in my postdoc. Really the first 2.5 years were spent characterizing resident memory T cells that populate the lung draining lymph node after a flu infection. And so resident memory T cells, a great feature of resident memory T cells, they're remarkably long-lived, but the exception to this really seems to be the lung. TRM in the lung don't seem to stick around for very long, and that's been a problem for vaccination strategies that have targeted TRM in the lung. And there's been several explanations to sort of explain the relative short-livedness of lung TRM. So in 2020, we were able to demonstrate or provide evidence for an additional mechanism, which we refer to as retrograde migration, whereby cells that have committed to a residency program in the lung nevertheless egress through the draining lymphatics for more permanent residence in the draining lymph node. I'll mention briefly that this is a cartoon that I made for the cover of JAM. Unfortunately, they went in a different direction. But I make all my own scientific artwork and illustrations, and that's been sort of a nice way to collaborate with people over the years in terms of review articles and grants as well. And so when the pandemic hit, I was able to sort of parlay some of the expertise that I had gained over the first 2 years of my postdoc to contribute to 2 papers that were published in JI on COVID that were both amongst the top 20 most read in 2020 and 2021. So now to jump back into sort of the main focus of the talk, TRM in the oral mucosa. So why study TRM in the mouth? As I've alluded to so far, our lab, the Massivist lab, currently tends to think about TRM in the context of viral infections, but TRM can really recognize and organize immune responses against a host of other insults, intracellular bacteria, dietary and environmental antigens, tumor antigens, and self-antigens. And because of this, TRM have been shown to play a really important role in several human health and disease states. And while TRM have really only been relatively recently characterized, their tissue-specific functions have been defined quite nicely in places like the gut, the urogenital mucosa, the skin, and the lungs. Immunologists have by and large avoided the oral mucosa as a place for studying TRM. So at present, there really is a fundamental void in our understanding of the ontogeny and functional implications of resident memory T cells in the oral mucosa. And I think there's sort of 2 main reasons for that. The mouth is kind of a difficult tissue to isolate. You can take out a pair of lungs, you can take out a gut, you can't really take out a mouth. It needs to be sort of delicately dissected from the underlying muscle. And then related to what I'll get into in a few slides, there's presently a lack of animal models for generating large quantities of resident memory T cells in the mouth to manipulate and study. So these are some of the obstacles that I had to sort of overcome with this project. But why even have an interest in TRM in the mouth? I think that as this project has developed, I've gained a much greater appreciation for the mouth. The mouth is a very important organ. Obviously, it initiates alimentation. It's chemosensory. It's used for verbal communication and nonverbal communication. It's very much tied to our personality. I think that became painfully evident after 2 years of mask wearing. But the mouth is also highly susceptible to disease. So periodontal disease is the world's most common chronic inflammatory condition. And the mouth is, of course, also susceptible to oral infections like HSV and HPV. Oral cancer is the 6th most prevalent cancer globally. And, of course, there are oral manifestations of autoimmune disease, the classic being, xerostomia associated with Sjogren's syndrome. And at least with respect to the first two here, periodontal disease and oral infections, historically, efforts to sort of prevent or mitigate these diseases have focused on the microbiota. So deal with the infection, deal with the microbiota. In the case of periodontal disease, you'll deal with the problem. I think more recently, there's been an interest in targeting specific immune cells and or pathways as a novel means of treating oral disease. And here I think that resident memory T cells may be relevant. Two examples where I think TRM could be playing an important role. I'll highlight periodontal disease and oral cancer. So periodontal disease is a progressive disease that progresses through these various stages. First, gingivitis, which is a reversible stage of the disease, and then you get into the irreversible stages of disease, which are defined by pocket depth, which is essentially the degree by which the infection and corresponding inflammation has eroded the dental supporting structures in alveolar bone. So it's thought that this process is initiated by dysbiosis within subgingival dental plaque biofilm communities. And that dysbiosis triggers an immune response that essentially goes unresolved. And over the course of this chronic disease, this persistent immune response to this infection really results in this collateral damage. And so these supporting structures, the cement and the alveolar bone, they literally hold the tooth in place. And as the disease progresses, the grip that those structures have on the tooth weakens and weakens, and ultimately the tooth is exfoliated. And so, anecdotally, many patients with progressive forms of disease have been shown to have active oral viral infections, suggesting there could be some viral etiology to this disease. Cells bearing a residency phenotype, that is CD103 positive, CD69 positive cells, have been shown to be enriched in human periodontitis. Antiviral therapy has been shown to mitigate disease. Again, all of these sort of point to a potential role for antiviral T cells as maybe an initiator or driver of disease. So in the context of periodontal disease, oral TRM may be playing a pathogenic role. But the flip side of that coin is of course, the context of oral cancer, and this is work quite relevant to the work being done by Pam Rosado here. So this is phenotyping CD8 T cells from a human oral squamous cell carcinoma that I helped Pam process, and you can see here in this tumor that there's a large population of CD8 T cells that bears a residency phenotype, so expresses CD103 and CD69. So these TRM are in human oral cancers, and of course, a question is, can we leverage the immunostimulatory potential of TRM in oral cancers as a novel, inexpensive, and non-invasive oral tumor immunotherapy? So a lot of parallels there with what Dr. Rosado was doing. So the initial questions that this project wanted to address is, first of all, do CD8 TRM populate oral tissues following a systemic viral infection? That's kind of, these systemic viral infections are sort of the wheelhouse currently of the lab. And if so, can we manipulate their abundance in the mouth? And lastly, for this initial part of the talk, what are what oral compartments, what tissues in the mouth do oral TRM reside? Now, the mouth is sort of a broad statement. The mouth comprises many architecturally distinct tissues and microenvironments. There's different layers of stratification and keratinization and oxygen and nutrient levels. And so, to say the mouth is maybe a bit simplified. So, what specific compartments do resident memory T cells reside? Before I jump into the data, I just want to introduce how I isolate murine oral tissue. This is a three-step process. It first involves removing the head and immobilizing it on a dissection board. We can then make an incision through the mandibular incisors and sort of exposed the oral mucosal lumen using bent 18-gauge needles. And so here we can isolate the various tissues that we typically collect. So the tongue in green can be easily removed, and then we can, of course, isolate the gingiva and the buccal mucosa, or the cheek. So I'll use those terms interchangeably. Buccal mucosa is the cheek, shown in blue, and then, of course, the hard and soft palate in yellow. And this is what that looks like before and after tissue isolation. So the first question we wanted to ask was whether or not a systemic viral infection seeds the oral mucosa with antiviral TRM phenotype cells. And so for this initial question, we compared two sort of commonly used systemic viral infection modalities, lymphocytic meningitis virus, or LCMV, and vesicular stomatitis virus, which expresses OVA, or VSV OVA. So for these experiments, we utilized transgenic CD8 T cells. So P14 T cells recognize the GP33 epitope from LCMV, and OT1s, of course, recognize ovalbumin, which here has been engineered to be expressed by VSV OVA. So for this setup, we transferred 50,000 of either P14 or OT1 T cells intravenously 1 day prior to a systemic infection with LCMV or VSV OVA. And at memory time points, I believe this was 80 days post-infection, we enumerated transgenic CD8 T cells in the mouth, and what we found was that VSV OVA was superior to LCMV at generating memory T cells in the oral mucosa. So in white are enumeration of cells generated from LCMV, and in blue is the enumeration of OT1s generated through the VSV OVA infection. And you can see that there are more cells in the oral mucosa after a VSV OVA infection, and if you look at the phenotype of those cells, they express a residency phenotype. They're 103 positive, Ly6C negative, or tend to be mostly 103 positive, Ly6C negative. They also express CD69. And so the conclusions here was that just comparing these two systemic infection modalities head to head, VSV OVA was superior to LCMV. Many of these cells, as I mentioned, expressed canonical residency phenotype. So in the interest of time, I just want to summarize a bit of data that we sort of followed up after doing these initial experiments. So we utilized parabiosis surgery to confirm that most of the memory T cells in the oral mucosa after either LCMV or VSV OVA infection were in fact resident. But we followed up and did subsequent experiments with VSV OVA simply because it generated more cells in the oral mucosa than LCMV. And so we did some protection experiments. So we showed that mice that had expanded OT1 T cells through VSV OVA infection, if we challenged those mice with a novel viral infection that expressed a shared CD8 T cell epitope, in this case vaccinia virus expressing OVA, if we infected the underside of the tongue of these VSV OVA immune mice, that those mice were protected from infection. And this is sort of the strategy that we use to do these lingual infections. So I built a mouse restrainer, and we basically take a tattoo needle and we dip it in neat vaccinia ova stock, and then we use that to penetrate the underside of the tongue, which is sort of relatively thin epithelium and non-keratinized. And so we showed in this context that mice that had previous VSV OVA infection were protected completely. So suggesting that VSV OVA infection generates protective, antiviral TRM. And we utilized FTY720, so this is a reagent that will sequester circulating cells within lymph nodes and spleen to show that when we took away the circulating immune component, we did not compromise protection to this lingual VAC-B OVA infection, which is compatible with the interpretation that local TRM were conferring protection in this model. So the systemic viral infection generates TRM in the mouth, and they are protective. So you may have noticed when I was showing you a head-to-head comparison of LCMV and VSV OVA that even the superior systemic viral infection model, VSV OVA, only generated about 300 cells in the oral mucosa. And we figured that could be improved upon. It's sort of hard to build a research program around 300 cells. And so we devised the strategy we're referring to as a VPEP, which stands for viral prime epitope pull, and I'll refer to it as VPEP throughout the rest of the talk. So the strategy addressed the additional question of whether local antigen recognition would potentiate oral TRM formation. So what we did here is, sticking with VSV OVA, again, we transferred OT1 T cells into congenically mismatched recipient mice so that we could track this OT1 population. 1 day later, we infected systemically with VSV OVA. But here, sort of 5 to 6 days later, during the clonal expansion phase of the immune response to VSV OVA, we anesthetized mice and we swabbed their oral surfaces with a fine hobby swab that we dipped in a slurry of dental pumice. So this is a mechanical irritant and abrasive. This contraceptive gel that contains an active ingredient in inoxanol 9 or N9. So this is a surfactant. It's an epithelial disrupting agent. And then within that slurry of irritants, we had included synthetic peptides, so cognate antigen that the OT1 T cells would recognize. So the idea is that during this clonal expansion phase, OT1s are promiscuously seeding tissues, including the oral mucosa, but now they're going to see a second hit of antigen in the mouth directly in the context of irritants. And the hope was that the abrasive agent and the epithelial disrupting agent would help tease apart the epithelium to allow the synthetic peptide to sort of penetrate deeper into the tissue where these OT1s could recognize that peptide. And hopefully we would establish a larger population of TRM to study. I don't know why my thing's not advancing. Okay, this just maybe times out after a while. So here is data showing that this VPEP strategy works. So here I'm comparing mice that received a systemic VSV ova infection alone. So again, in the previous slides that I showed, this generates about 300 cells in the oral mucosa. In red are mice that received this VPEP strategy, so systemic VSV ova infection alone, 6 days later swabbing with irritants containing the relevant synthecopeptide. And in gray, we swapped out the synthecopeptide with an irrelevant GP33 peptide. So these mice are getting a systemic viral infection. They're getting orally swabbed with irritants, but in those irritants is an irrelevant peptide that would not, that OT1s will not recognize. And so this is one of those examples where something worked pretty well the first time and we kind of ran with it. So you can see that our VPEP strategy, we get, we go from getting a couple hundred cells in the oral mucosa to around 1,000. This represents a 4-fold enhancement of OT1s in the mouth that we can generate and isolate for flow cytometry. And then importantly, if we had swabbed the oral surfaces with irritants containing an irrelevant peptide, we don't see enhanced oral TRM generation above a systemic VSV ova infection alone, which tells us that it's not simply the act of swabbing and injury associated with that swabbing that is helping establish a larger population of TRM in the mouth. So here then the conclusion is that this VPEP strategy is superior to VSV ova, and that this VPEP strategy is an antigen-dependent process. And without showing the data, I'll just mention that we've then subsequently performed parabiosis experiments on VPEP memory mice to show that upwards of 95% of the OT1s in the mouth after this VPEP strategy were in fact residents, and most of them, upwards of 75 to 80, express CD103. So I'll utilize these VPEP mice for the remainder of the talk. So where are these cells located? So up until now, I was isolating tissue, and I should have mentioned when I showed the process for isolating oral tissue is that I typically pool those tissues into a single oral mucosal sample that then gets processed for flow cytometry. So when we do that, of course, we lose any information about the spatial distribution of cells in the tissue. So we wanted to ask, where were these cells in the tissue? And the sort of the punchline here is that they're really everywhere. So this is projecting quite well, I think. So in white are our VPEP elicited OT1 TRM. So in the palate, you can see that they're broadly distributed. This sort of arrow represents the transition between the hard and the soft palate. The tongue, their sort of distribution was a little bit more localized. We find a lot of cells in the underside of the tongue, and we find a lot of cells associated with mixed serous and mucus glands, the posterior dorsum. However, there were several cells sort of scattered throughout the tongue as well. And this is just showing highlighted our magnified images. We see them throughout the cheek. They're associated with the sort of the basal epithelial layer in all of these locations, and we also find them in the gingiva. So you can see this beautiful dovetailing of the gingiva that gives us the masticatory mucosa its strength. I should mention that the gingiva, in terms of epithelial tissues in the body, is probably the one that's subjected to the most intense mechanical stress because, of course, we chew and the masseter is the strongest muscle in the body. So this needs to be an incredibly resilient epithelium. And so we find cells in the gingiva. The gingiva is sort of difficult to image by conventional anatomic microscopy because it is such a small piece of tissue. So we utilize some sort of maybe more complex imaging techniques. Here we've decalcified and cryo-sectioned an entire upper jaw. And so you can see this is sort of interproximal gingia here. And this is where the maxillary first molar would be, but it's this is decalcified, so the tooth is no longer there, but you can see in white that there are cells, sort of within these the dental supporting structures that surround the tooth. And again, if you're interested in gingivitis, which impacts these structures, these cells are in the relevant location. We utilize clear enhanced three-dimensional imaging to get a better picture of TRM in the gingiva. Again, this is after our VPEP strategy here. Our VPEP elicited TRM are in green. And this is sort of the analogous location from the mouse and the human mouth, the mucogingival junction. And so you can see in this three-dimensional rendering that this is what sort of the tissue looks like, but in a two-dimensional slice, you can see that there's a high density of cells in the gingiva. So again, this is an incredibly small piece of tissue, but despite its small size, there's a high density of these antiviral T cells in that location. We also see them in other interesting, potentially hypothesis-inspiring locations. We find them in bone marrow chambers in the lower jaw. This is what they look like there. This is an entire decalcified lower jaw mandible. So the lower incisors, mandibular incisors, would extend out this way, if you can imagine here. So we're looking at the side of the teeth, sort of from the tongue's point of view. And the interesting thing here in these, it's sort of probably hard to see, but we in fact, we find TRM within the pulp of the tooth, which I think is also quite interesting. And we also find them within the mandibular molar roots, again, in these structures that are really holding the tooth in place. These antiviral T cells are present. And then lastly, we looked at the underside of the tongue and the floor of the mouth. So this is an entire decalcified mandible again, and here we're looking at the submandibular duct. So if you're interested in saliva and saliva production, I think this is also kind of hypothesis inspiring. We see this as the duct itself, and you can see that this structure is full of antiviral T cells. So these VPEP-elicited TRM are really broadly distributed throughout the mouth, and they're in locations that are relevant to periodontitis, to saliva production, as well as I'll talk in the end, potentially taste. So what I've shown you so far in the first kind of half of the talk is that systemic viral infections are capable of generating TRM in the oral mucosa. I didn't show you this data, but we did experiments to demonstrate that oral TRM appeared to be sufficient for protection to subsequent viral infections that express shared CD8 T cell epitopes. Our VPEP model is now a great tool for generating large populations of TRM in the mouth, and it additionally shows that local antigen recognition really helps generate abundant populations of TRM in the oral mucosa. And oral TRM generated through our VPEP strategy are really broadly distributed.
Mike?
Yep.
Question. Have you thought about what happens if you have local priming, right? Because, I mean, that's the real purpose of resident memory, right? You get primed in the oral here or the lungs, and you're only showing kind of the secondary hit being locally.
Yeah. We tried to do a primary infection with vaccinia, with VSV ova, and we didn't get a very productive infection, but I think I haven't tried transferring OT1s and just giving the peptide, and that might be an interesting, way to address that.
You looked whether VSV antigens in the tongue, right, that has some promiscuity to tissues and hanging around long term.
We haven't. The S in VSV is stomatitis, which is an oral ulcer that cattle apparently get when they're infected. So there probably is some tropism for the mouth, but we should look. Yeah, but good questions. So moving on. Of course, we wanted to address how what happens when oral TRM that are established through this VPEP strategy or elsewhere are what happens when they're reactivated? How does that impact oral immunity? And can we develop strategies for eliminating oral TRM once they're established? And finally, if we could develop a strategy for depleting oral TRM, would that abrogate inflammation in the presence of cognate antigen? I think it's time that again. Oh, I gave away the punchline here. Okay. So to address the first question, what are the consequences of oral TRM reactivation? I think this kind of now gets into the part of the talk that I'm really excited about. So we wanted to address whether or not oral TRM reactivation would recruit bystander memory T cells into the oral mucosa. And so the experimental strategy here is shown on the left. So we generated our VPEP memory mice as I described. At 30 days later, we split those mice into two groups. Both groups received a memory CD8 T cell population that would have antigen irrelevance. In this case, we transferred in LCMV specific memory P14 T cells that were additionally marked with a congenic Thi 1.1. So we're transferring a memory CD8 T cell population that in this context will be irrelevant for the antigen that we're using to reactivate TRM. And so those mice then, control animals were orally swabbed with the pumice and N9 alone. So a sham swab here, and then the experimental group of mice received oral swabbing with synthiclopeptide dissolved in irritant, so the TRM reactivating peptide. So we're really asking in this experimental setup, does reactivation of oral TRM that are established through VPEP recruit a circulating bystander memory CD8 T cell population into the mouth? And we used histology to address this question. So in the control swab mice, we find a smattering of OT1s. These are depicted in red. But in all of the tissue sections that we looked at from the multiple mice that were used in this experiment, we'd never found a single bystander recruited P14 in the oral mucosa. And here I'm focusing on the cheek. If we reactivated TRM in this context, we now see them. I'm highlighting several examples here that we see recruitment of those bystander cells into the cheek. And importantly, they're not broadly distributed in the cheek. They really tend to associate or aggregate with presumably reactivated OT1 T cells. So here I've just removed the nuclear and ECAD staining so you better visualize these, what we're sort of referring to as inflammatory clusters. So we're able to get bystander recruitment. So we were interested in sort of what might be the mechanisms that were driving this recruitment, and just broadly what happens when oral TRM reactivated transcriptionally. And so we performed a bulk RNA sequencing analysis. So in this experiment, we took VPEP memory mice. Those mice were split into three groups. One group was left untreated. A second group received oral swabbing with an irrelevant peptide, in this case GP33 again. And then the third experimental group received oral swabbing with TRM reactivating synthiclopeptide. 12 hours post-swabbing, we isolated the buccal mucosa and submitted that for bulk RNA sequencing. And so PCA analysis revealed that TRM reactivation was driving transcriptional changes, although we saw a small adjuvant effect in mice that just received the swabbing alone. And I think that's good because it shows that the irritants are actually doing something. So when we look at the volcano plots, we see the relevant comparison here is comparing the sham swab to the TRM reactivation swab groups. And we see this sort of eruption of almost 900 genes. If we look at individual genes, we see some of the usual suspects, things you might see when you get reactivation of an antiviral CD8 T cell population. So there's a strong induction of interferon gamma, granzyme B, perforins. But we also saw induction of several CXCL and CCL chemokine genes, which inspired us to sort of return to a histological approach to look at whether or not reactivation of TRM recruited or reorganized kind of panleukocytes in the oral mucosa, again here focusing on the cheek. So this is 8 hours post-swabbing with either a control peptide, GP33, or I'll show you the synthiclopeptide. And again, this is looking in the buccal mucosa. And so in the control context, you find a few OT1s, and there's some 45 positive cells. Again, this is a panleukocyte marker. We're sort of limited with the number of colors that we can use for IF microscopy. So this is sort of a broad pass here. But I think what was really nice is when you reactivate TRM by swabbing with the relevant synthiclopeptide, you get these massive inflammatory clusters. And I hope that you can see that within these clusters of inflammatory cells, pan-CD45 positive cells, you see these presumably reactivated CD8 TRM. And so we saw this in the cheek. I think it was also really quite interesting if we looked in minor salivary glands in the buccal mucosa. We see again in the context of a GP33 swab, you find these nice salivary glands, minor salivary glands that have this nice sort of acinar morphology, but there's not a lot going on with respect to inflammation in that tissue. If you reactivate TRM, you now get this massive salivary gland inflammation. So if you're interested in salivary biology like I am, you see this, and that sort of is exciting and may have relevance for a disease like Sjogren's syndrome. So looking a little bit, taking a deeper dive into some genes that may be relevant for oral physiology and disease. And I'm sorry, there's a lot of information here, so I won't go through individual plots, just to sort of highlight general themes that we're seeing in our sequencing analysis. In white is no treatment, in gray is treatment with the relevant GP33 peptide, and in red is swabbing with the relevant synthiclopeptide. what we see is that when we reactivate TRM and look at the sequencing, we see induction of several toll-like receptor genes, really strongly induced when TRM are reactivated. We also see genes that are induced relative, or that are involved in keratin production and mucin production. So that's quite interesting from thinking about reinforcement of the oral tissue and also the composition, the mucin composition of the saliva. We see genes relevant for opsonization and ADCC, several Fc gamma receptors. We also see a neutrophilic signature, and I'm just highlighting here specifically genes involved in calcium and iron sequestration, which are important cofactors for the oral microbiota specifically. And we also see complement genes strongly induced, and I think also quite interestingly, we see induction, albeit it's subtle, of the polymeric Ig receptor, which potentially suggests that there's more transcytosis of mucosal IgA into the oral lumen when TRM are reactivated. Collectively, this kind of inspires the hypothesis that TRM reactivation, although we're reactivating an antiviral population, we may be impacting the oral microbiota. Again, we're seeing heightened sensitivity to the microbiota, neutrophilic signature, complement, and the like. Sorry. So we looked at some other genes that we thought might be interesting and relevant, we see several genes induced that are related to mucosal sensation, some temperature, some pain, specific genes. But I want to sort of point your attention to some of these genes down here. So despite the fact that we're isolating the buccal mucosa, which isn't directly involved in a disease like periodontitis, we're seeing genes strongly induced that are known to be etiologic in periodontal disease. So we see upregulation of several matrix metalloproteases, MMP3, 8, 9, and 25. And I should note that MMP 8 and 9 are the two most relevant MMPs in periodontal disease. And we also see genes involved in bone resorption and pyroptosis, which have also been sort of implicated as drivers of periodontal disease. So our sequencing analysis was a bulk sequencing analysis. We're swabbing with peptides to reactivate TRM, but it's possible that we're reactivating effector memory T cells that are transiently traversing the tissue, or perhaps there's some other T cell reactivation going on. So we really wanted to hone in and ask, what is the consequence of bona fide TRM? And we know based on our parabiosis experiments that TRM in the oral mucosa after our VPEP strategy expressed CD103. So we thought of it, or we wanted to devise a system where we could deplete 103 expressing TRM in the oral mucosa. So we came up with this strategy, and I'll walk you through the experimental design because I think it's a clever approach, but it's maybe a bit complicated. So the design here is that we would transfer OT1 T cells and expand them through our VPEP strategy in a CD103 deficient host mouse. Because this OT1 that we transferred is capable of expressing CD103 when they get into the oral mucosa after VPEP, these OT1s would be the only cell population capable of expressing CD103 in the 103 deficient host mouse. So then we figured that we could use antibodies to target 103 expressing OT1s, and there wouldn't be any off-target effects of that antibody treatment. Again, because the host mouse is CD103 deficient. And to make a long story short, conventional antibodies did not end up depleting our OT1s. It simply blocked ex vivo flow cytometry staining. But luckily, we found a company that manufactures this toxin conjugated anti-CD103 antibody. So this is saporin toxin, which is derived from the soapwort plant. This is a ribosome inhibiting protein, and the acronym is perhaps fitting because the idea is that when this molecule binds to a cell that expresses CD103, it kills the cell. I may just stay down here for the rest of the talk. So what we found was that this depletion strategy worked quite nicely. So here we're looking at 103 expressing VPEP elicited TRM in the oral mucosa in mice that received saporin conjugated IgG antibody versus the saporin toxin conjugated anti-CD103 antibody. And you see that we're able to get upwards of 90% depletion of 103 expressing TRM in the oral mucosa, whereas the 103 negative subset is unaffected. So we had a really nice system here for eliminating 103 expressing TRM. So with this system then in place, we returned to our bulk sequencing and asked which of those transcriptional changes that we,
Quick technical question. Do you get equal priming because you don't have the CD103 dendritic cells?
We haven't looked head to head. There does seem to be kind of a muted response in these mice, just in terms of the Excuse me. So I'll show the sequencing data. In the wild type context, when we reactivate TRM, we get about 900 genes that are strongly upregulated. Almost all of those genes they're also upregulated compared to the control mice, but they're not quite as strongly upregulated. So I think there is probably a bit of an impact of not having 103 expressing DCs or other subsets.
And do you get any CD4s into the tissue? Because they may need it to be retained in the oral mucosa as well.
Yeah, haven't looked, but we should look at that. And it's possible that maybe there's regulatory subsets in the oral mucosa that are expressing 103 and are lacking 2 in the 103 deficient mice. But it is so it is kind of a contrived system. But I guess I should mention that most experimental strategies for addressing the role of circulating versus tissue resident memory T cells have targeted the circulating population. So using Thy1 depleting antibodies or other approaches. This really, to our knowledge, is the only system that we have to eliminate the TRM while preserving the circulating population. So I think it's kind of a neat tool and hopefully would be relevant to people in the room. So we return to the sequencing experiments utilizing 103 deficient host mice in which we induced this VPEP model. And again, mice got either the relevant control antibodies or the anti-CD103 conjugated saporin antibodies. The system here is maybe a little bit complex, but essentially we had 3 groups. We had a group of mice that received a control swab with GP33 peptide, a group of mice that received a TRM reactivating swab with sympecopeptide. Both of these groups of mice received the saporin conjugated IgG antibody, so all of the mice got saporin toxin. Then the experimental group was depleted of 103 positive TRM 1 week prior to reactivation with the relevant TRM reactivating peptides, sympecopeptide and irritants. And then 12 hours later, we submitted buccal mucosa again for bulk sequencing. So here we can see really nicely that these sort of plots are all relative to the control swab mice, the GP33 mouse, that when we reactivate TRM, again, we see induction of a number of genes, in this case 177 genes are significantly induced, and almost every one of those genes is significantly blunted when 103 positive TRM were depleted prior to swabbing with sympecopeptide. And sort of the median values are shown here for these 177 genes. More impressively, there were 350 or so genes that were highly induced upon TRM reactivation that were completely blunted when 103 positive TRM were depleted prior to symphecal swabbing. So we see here on the sort of the heat maps some representative examples of genes that I've shown before. Interferon gamma, granzyme B, for example, are induced upon swabbing with an intact TRM compartment, and that those genes are strongly reduced when 103 positive TRM are depleted. Additionally, there were 80 or so genes that were significantly downregulated when TRM were reactivated that were now re-expressed or fully restored when 103 positive TRM were depleted prior to swabbing. And interestingly, and maybe relevant to the chalk talk tomorrow, several keratin genes were actually downregulated when TRM were reactivated, and many of these genes were restored when 103 positive TRM were depleted prior to reactivation, suggesting that although there's a clear anti-pathogen response going on when TRM reactivated, there may be it may be compromising the integrity of the oral epithelium. We also saw genes that were relevant to digestion, interestingly enough. So amylase, for example, was down when TRM reactivated, and it was slightly more expressed when 103 positive TRM were depleted prior to swabbing. So what I've shown you then in the second part of the talk is that TRM reactivation mobilizes circulating cells into the oral mucosa, so we can show bystander recruitment. TRM reactivation drives transcriptional changes in the mouth. We've seen evidence that panleukocytes reorganize and accumulate around putatively reactivated oral TRM. Genes involved in microbial sensing and periodontitis are also strongly invoked upon oral TRM reactivation. We have the system saporin 103, which is a novel tool for therapeutically eliminating 103 expressing TRM from the oral mucosa. And lastly, we showed with the bulk sequencing data that if we deplete 103 positive TRM prior to swabbing, we can abrogate transcriptional changes. So I'll just close with sort of a summary of the tools that I've developed and sort of the future research directions that my independent laboratory will focus on. So, really, I think with this VPEP strategy, we have a nice system that we can generate large populations of TRM in the oral mucosa, and we can also use that same strategy to temporally reactivate them, whenever we choose. So we can make them, we can reactivate them, and then we can deplete them with the Saporin 103 reagent. Additionally, in my postdoctoral experience, I've developed tools and techniques and a skill set for isolating oral tissue, for imaging the oral tissue. We have lingual infection models going, and I'll talk a little bit about experimental periodontitis. So really the three main thrusts of my independent research laboratory will be addressing these questions. First, does oral tumor reactivation exacerbate ligature-induced periodontitis? This is a mouse model of periodontal disease. Second is chemosensory biology impacted by oral tumor reactivation. I'll show you some data that might support that aim. And lastly, can we leverage the pro-inflammatory functions and potential of TRM as a novel anti-oral tumor immunotherapy? So this will be kind of the focus of my chalk talk for tomorrow. So I'll just kind of show some preliminary data which will be relevant for tomorrow. So the model of periodontal disease in mouse is that's commonly used is this ligature-induced periodontitis model, where a ligature is placed around the maxillary second molar of mice, and over the course of a week or so, it's thought that this ligature causes dysbiosis that then drives an inflammatory response that results in bone resorption. And this is what this looks like at day 8. And so we can image this various methods, but we typically use micro-CT imaging. And so, the questions we want to address is, if we place ligatures concurrent with oral tumor reactivation, will that exacerbate disease? And we can ask questions about inflammatory infiltrate, the microbiota, and disease resolution.
Can I ask a question about this here?
Yep.
So, in human periodontal disease, the root cause is thought to be bacterially driven dental plaque.
Mm-hmm.
So do the mice have plaque, and can you measure it? And if it's really microbially or bacterially driven, it sounds like the TRMs then they're probably largely antiviral.
Yep.
So is this all secondary? And so have you done things like do this in notobiotic mice, or give them anaerobic antibiotics that deplete their oral flora?
So we haven't, but these will be sort of aims of my chalk talk. So yeah, we do want to say, can we take away the microbiota component? If we deplete the microbiota, in a notobiotic setting, which it wouldn't be depletion, or with broad spectrum antibiotics, so we take away the microbiota, can we reactivate TRM in antiviral population and still get evidence of bone loss?
Yeah, you wouldn't need broad spectrum antibiotics, largely just anaerobic.
So yeah, that's great. But yeah, those are questions that I'll talk about tomorrow during the chalk talk. So this is what a ligature looks like when it's placed in the mouse. So the first molar in mice is the largest molar if things look a little wonky to you. So this is what it looks like in a ligature that I placed, and I show this image because this is the image from my title slide. This is the exact location where this ligature is placed. This is the maxillary second molar. This is just a baseline in a VPEP mouse. But you can see VPEP elicited TRM in white. So these cells are really at the scene of the crime with respect to periodontal disease. We've done some very preliminary bulk sequencing analysis on reactivated gingiva, and we see, again, compared to the mice that received the control swab, we see really strong induction of several effector molecules, chemokines, genes relevant to periodontitis and complement. And we've started to do some ligature-induced periodontitis experiments. And so in this experiment here, we take our VPEP memory mouse, we swab with either a control peptide or some pecylpeptide and irritants, and immediately after swabbing, we place ligatures. 4 days later, we gave an additional swab with either the control peptide or the pecylpeptide. And then 3 days later, so this is ultimately 7 days post the initial ligature placement, we imaged the maxillas and quantified bone loss. And what we're seeing is quite promising, that we are seeing that when you reactivate TRM concurrent with ligature placement, you're able to further drive the disease. And then we've also done some characterization of cellular infiltrate into the gingiva after ligature placement. In these experiments, ligatures are actually placed on both maxillary molars so that we can maximize the tissue that we can isolate cells from. And we see, strong recruitment of OT1s, but also strong neutrophilic signature. Monocytes and macrophages are also recruited into the gingiva. And then histologically, if we look at gingiva in a mouse that receives a control swab, you see OT1s, but not a whole lot going on in terms of pan-CD45 cells. If TRM are reactivated with synpecylpeptide, you now see recruitment of a large population of CD45 positive cells into the gingiva. And if we use our Saporin 103 depletion system to deplete 103 positive oral TRM prior to swabbing with the reactivating peptide, we mitigate that inflammatory response. So this all really suggests that TRM can drive this oral inflammation. A few more minutes here. I just want to touch on some work that Yina alluded to, looking at chemosensory biology. So another interesting thing to come out of our sequencing analysis is that we saw that when TRM were reactivated, in the red bar here, we saw induction of some olfactory receptors. This is olfactory receptor 56. This is a receptor for limonene, which is a component of citrus fruit peel oil and a flavoring component of several lemon lime soft drinks. In the context of the system where we can reactivate TRM in the context, or reactivate CD8 T cells in the context of 103 positive TRM depletion, we see that when the 103 positive oral TRM population is intact, we again see induction of olfactory receptor 56. If the 103 expressing TRM are eliminated, we abrogate that response. And so, this really begs the question: do mice with reactivated oral TRM prefer Sprite to Pepsi? This is only slightly ingest. It really kind of gets to the bigger question of whether or not oral TRM reactivation can impact sort of the behavioral responses to food. So in mouse, the taste buds and taste perception is a little bit different in humans. Sort of the tongue and the soft palate really represent the primary taste sensing locations in mice. And in the tongue, you have the folate, circumvallate papilla, and of course, spongioform papilla throughout the dorsum of the tongue. But I just want to highlight the structure of the circumvallate papilla is at the back of the tongue. And that there are also a number of isolated taste buds in the soft palate. So why, again, am I talking about taste here? If we look in our VPEP memory mice, here at the soft palate, we find that these structures are being actively surveilled by TRM, so they're shown in red. So this is just two examples of the soft palate. Of course, I could have cherry-picked examples. Here's looking at clearly enhanced three-dimensional imaging of the soft palate. This is the 8th palatine ridge, so the transition between the hard and the soft palate. And you can see here that these structures here are taste buds. These are likely salivary ducts. But if we zoom in here, we find that OT1s, our VPEP-elicited TRM, are in pink. And here, looking through various view stacks, you can see the characteristic crescent moon morphology of these taste buds, and you can see that these structures are being surveyed by TRM. This is the circumvallate papilla, the back of the tongue. This is what it looks like under the microscope. So we've looked at this structure histologically. This is in a mouse that receives a control swab. This is the circumvallate papilla. You can see that there are a few OT1 T cells in the structure at baseline. When you reactivate those cells, you get massive inflammation in this circumvallate papilla. If 103 positive TRM were depleted prior to swabbing the reactivating peptide, you mitigate that response. So, this inspired us to look at reactivated circumvallate papilla in soft palates. And just in the interest of time, we're seeing that there's induction of bitter receptors. There's about 38 bitter receptors in mouse and humans, and we see evidence that if you look at pan-bitter receptors, there's about a threefold enhancement in potentially the perception of bitter in mice with reactivated oral TRM. The last slide here, before I might give my acknowledgement slide or my thank you slide, is again to look at the potential for oral TRM reactivation as a novel oral tumor immunotherapy. This is a clear enhanced image of the underside of the tongue, but you can see that the high density of TRM generated through VPEP. So a question, of course, is we're planning on using a chemically induced oral cancer model. This is for NQO. This induces oral squamous cell carcinomas. So the question is, if these tumors develop in a VPEP memory mouse tongue, and the tongue is a primary location where tumors develop in this model, will those tumors incorporate TRM? Can you reactivate TRM by swabbing or injecting the tumor with peptide in the context, or alone or in the context of checkpoint blockade inhibitors? And will that curtail tumor growth? Again, this is very similar to some of the work that Dr. Rosado is doing. So with that, thank you for your time. I've left a few minutes for questions, but I can also stay a little bit later if anyone wants to follow up. Thank you very much. Yes.
So obviously, kind of rodent mouths are different to human mouths, and one of the differences, at least, is that the rodent teeth keep growing right, throughout life.
Yep.
And I just wondered if you thought that had any impact on the on like the recruitment or retention of TRM? Because it seems like they're really proximal to the teeth, right?
Yes. So yeah, as I understand it, it's just the incisors that are continually erupting and not the molars. But yeah, it's an interesting question. One of the genes that we did see in our sequencing analysis was this SSU2, which is a gene that's important for odontogenesis, so for tooth growth. So we see that it is actually downregulated when TRM are reactivated and is restored when 103 positive TRM are depleted. But yeah, in terms of the relevance there, we'll have to think about that a little bit. But yeah. Yes.
So in cancer treatment, patients get rank ligand antagonists and they get bisphosphonates to treat bone metastasis. Now, a big problem there is jaw osteonecrosis, completely unknown black box, big problem. This looks like maybe you've got the answer to jaw osteonecrosis. If nothing else, it's worth a lot of grant money because people really want to know what causes it. Have you thought about that?
I haven't thought about that, but like rank ligand is, of course, will I think accelerate bone resorption, and is a huge one of the main genes in periodontitis as well. So
You also get it with bisphosphonate use, which is not known to affect rank ligand signaling. So that there's something. So maybe TRM.
yeah, that's interesting. I mean, that could be a great avenue to explore. I'm not a dentist. I should have prefaced all this by saying that. So in terms of this has been a learning process for me throughout the last couple of years. But yeah, I mean that's great advice and would be a great angle for acquiring funding.
So we have a question in the chat.
Okay.
And that's from Lee Sung, who asks, so there's some computational methods that really allow you to detect T-cell receptor sequences from bulk RNA sequencing data. So he was wondering if, you might expect to observe, T-cell clonal expansion after TRM reactivation. I guess I would speak to also the fact that it's drawing in,
Yeah.
other effector T-cells into the oral cavity.
Yeah, that's a great question. Of course, we're reactivating, an antigen-specific population, so the T-cell receptor there was, but yeah, in terms of what other subsets, what other specificities are expanding would be really interesting. I mean, admittedly, we're doing bulk RNA sequencing, which is a pretty crude approach these days, and I would certainly hope to leverage the expertise of Lee and other people in terms of looking at some more advanced techniques for sequencing analysis.
Well, so I guess the follow-up question is, do you expect that there are regulatory T cells in the oral mucosa that actually restrict entry of TRMs or effector T cells in there? Because there's a lot of microbiota in the oral cavity.
Mm-hmm.
So.
Yeah, I would expect there to be TRM. We see, we don't get a ton of cells. The VPEP strategy allows us to get a lot more OT1s, but in terms of like fixing and perming cells for FOXP3 staining, we've just done really crude analysis, CD4, CD25. We see a large population of what look to be regulatory T cells in the mouse. So yeah, whether or not reactivation of the TRM and we have interferon gamma, but also type 1 interferons have been shown to negatively impact regulatory T cell proliferation. So there may be sort of a crosstalk there. Reactivation of antiviral T cells may be limiting or inhibiting the regulatory functions of the TRM. And so looking at kind of the Th17 Treg access in the context of TRM reactivation would be something that I think would be really interesting to look at as well.
Well, so I know that you didn't find in your model a lot of, TRMs in a homeostatic state, but, it certainly seemed like in your collaborative studies you saw TRMs in oral cancer, right? So I wonder if you looked at, let's say, a pet store mouse.
Yeah.
Would you see a lot of TRMs where they actually have all these recurrent infections and whatnot?
Yeah. So we've looked in pet store mice, and there's certainly a lot more 103 positive, CD69 positive CD8 T cells and CD4 T cells in the mouth. So we haven't done peribiosis. I haven't looked at yeah. I've actually done some peribiosis studies, and yeah, I mean most of the cells in the mouth are resident by peribiosis as well in the dirty mouse model. So, but yeah, the dirty mouse definitely has more TRM. So natural bombardment does definitely play play a role there too. So.
So, in your depletion model,
Mhm.
you still have a circulating memory component.
Mhm.
So, have you tried doing like a systemic rechallenging model where the circulating memory gets activated, and would you still see the inflammation in the mouth?
We haven't done that. That's a good question. Yeah, to just like inject some small amount of peptide, IP or IV, to see. Yeah, in terms of trying to equate that to what's like how do you match the peptide that's just being swabbed in the mouth versus given systemically, I guess would be a challenge. I think technically to try to find out a dose that would be equivalent, but it's something that we could look at too. We also have tools for depleting systemic cells while preserving the resident population, so we could kind of approach your question from the opposite angle, deplete the circulators, reactivate, and see if there's any difference there. And the presumption would be no, but if there was a difference, then that difference would have come from the circulating population.
All right, well, thank you so much.
Thank you.